Hi, everyone. I'm Josh, Marketing Director of the SideOut Foundation. I'm here with Anna, our Marketing and Communications Assistant. Joining us today are Drs. Lance Liotta and Dr. Chip Petricoin, who are co-directors for the Center for Applied Proteomics and Molecular Medicine at George Mason University. Lance and Chip lead our research team, SideOut's research team at Mason, working to find solutions that will extend the lives of those living with stage 4 breast cancer. And today they're here to talk about some awesome research that they're working on as it relates to COVID-19. Welcome, guys. Thank you for joining us. How's everything going on your end?
Yeah, things going good. We're working in the lab on this COVID research. We're excited about and hopefully starting in the next few weeks with some general lab reopening as the state and the universities bring in the governor's mandated processes.
Anna's actually going to kick us off today with the first question. So Anna, whenever you are ready, take it away.
Yeah, I think we'd just like to start with hearing a little bit more about y'all's COVID-19 research project.
We're studying antibodies that appear in the patient after they've had COVID. It's really important to understand how those antibodies develop and how they might be used to treat the virus. So we're developing new technologies for measuring the antibodies using both the saliva, simple collection method, and also looking in their blood. And we're looking at the antibody levels and see if the antibody levels correlate with how severe their COVID disease was that they had. And then we're also seeing if those antibodies, when we collect them from the patient, will actually kill the virus in the laboratory. And so in that way, we can not only understand how to measure whether somebody's been exposed to the virus and might have now antibodies, but we'll also be able to identify donors who could donate their antibodies, their blood, they've recovered from COVID, and then they now might be able to contribute to helping others to treat patients who do have COVID by donating their antibodies. And so we're developing new methods for both testing for COVID-19 antibodies, but also for identifying the super donors who would be, the new weapon against COVID. And so that's why we're very excited to continue this clinical trial. Eventually, we're going to do a widespread surveillance to see who has antibodies to COVID, because we can measure the antibodies much higher sensitivity than other methods. So even patients who have low levels of antibodies, we can find that out. So I think there's a great need and increasing need in the future to measure antibodies. And then if the vaccine comes out in the future, do you already have immunity, or do you have no immunity and you should take the vaccine? So a lot of exciting things are going to come out of this. And with my background as a diagnostic pathologist and Chip's background from the FDA and as a molecular biologist, together we work very synergistically on all the questions that come up about this project.
Yeah, we've been able to, one of the special abilities of our laboratory here, which we've definitely utilized and exploited for our SideOut funded efforts with metastatic breast cancer, our CLIA laboratory. We have a special laboratory component that's a CLIA accredited and CAP certified laboratory, and this allows us to basically run the assays that we're running for our SideOut Foundation funded metastatic breast cancer research. That laboratory ecosystem, if you will, infrastructure, we can then utilize that same infrastructure for this COVID research. And our ability to measure these COVID antibodies really grew out of our infrastructure ability. And also we've been working a lot in trying to identify new saliva and blood markers for breast cancer and for prediction on who will respond best to different cancer therapeutics. So we've grown this great expertise really in looking at saliva and looking at blood and developing diagnostic tests with this. And so we we had these special kind of attributes. We also had special biobanks. We have funded concussion research, for example, that we've been doing salivary concussion research. And those samples were all taken pre-COVID. So we have all these biobank of all these samples that were taken before COVID appeared. And these can provide tremendously priceless and valuable samples that we can then use to measure how accurate our COVID testing is. And so it's this collection of attributes, along with we have some really fantastic collaborators here at George Mason University who are world-renowned molecular virologists who've been studying coronaviruses for years. And we have a BSL-3 laboratory here that's one of the few in the United States where we can actually test with the hot virus in the BSL-3 lab how well these patients' antibodies, as Lance said, actually inhibit the virus. And so we have this really special kind of planetary alignment here that that we're really proud of, but also, you know, we're serious about exploiting for our COVID research.
Yeah, so it sounds like you guys are in a really awesome and unique position to be able to really make some meaningful contributions. That's awesome to hear. I was wondering if you wouldn't mind giving our viewers at home just like a little bit of a crash course on what exactly an antibody is and the importance of having antibodies in diseases.
When anyone is infected with the COVID virus, the virus damages cells and the immune system reacts to the damage of those cells by first sending immune cells in to fight the virus, but also it starts making antibodies. The antibodies stick to the virus, and the antibodies are proteins made by certain immune cells, and those antibodies are Y-shaped, and they go right, and if they stick on the spikes of the coronavirus, right on the tips of the spikes, those spikes are used for the coronavirus to attach to the surface of the cell and infect it. So if those antibodies fight the coronavirus by binding to the tip of the spikes, then it blocks the coronavirus from getting into the cell. And so, but it takes some time for the immune system to wake up to the fact that an infection is happening, to survey all the antibodies that it can make and see which ones work the best, and then it starts producing them in large numbers. So while the patient is succumbing to the symptoms in the beginning, the immune system is starting to fight and is making these antibodies, and they start building up over time. It first makes a kind of antibody called IgM that is more non-specific. It just is a general fighter, but it's not very specific and doesn't bind very tightly. Then it makes these other new kind of antibodies called IgA and IgG, and both in the oral fluid and in the blood, and they bind more specifically to the coronavirus. Then it also has immune cells that attack the coronavirus. So this takes time, and patients, depending on their underlying conditions, the antibodies may develop fast enough to help them get better really quickly, or it might be harder and harder for the immune system to fight the virus because the patient might have some underlying condition that makes them more susceptible to the infection, or there might be other reasons that we don't understand. But as the antibodies develop, then the patient is recovering, and we believe that they'll protect the patient from any further infection. We don't have, in our research, we don't see any direct evidence that somebody can get reinfected after they recover from the virus. But we do see so far that if a patient has really bad symptoms, then they have more antibodies in their body. And if they have milder symptoms, they have less antibodies in their body. It might be just because you need more antibodies to fight more viruses. So that could be the answer. But the antibodies are really key, because we could take those antibodies out of one patient and give them to another patient to help, like we used to do before we had the polio vaccine to help polio patients. And we can also study those antibodies and see, the best responders who got better really fast, or their antibodies kill the virus really well, what's the difference? Why are those antibodies better? If we study how they bind to the virus and how they inhibit it, we could say, well, maybe this gives us some ideas for a better vaccine or a drug, or we could mass produce these antibodies and use them as a treatment. So many exciting things can come out of this, and we are continually amazed by, the immune system and how it can recognize the virus, and then for most people, they recover really, really quickly, and the immune system is very, very powerful. So it makes us feel optimistic that as time goes on, a lot of people are going to maybe get coronavirus, but most everyone, except those who have underlying conditions, are going to get better and recover and not get it again. And particularly young people and children and athletes have, very mild, usually a mild course of disease, or really asymptomatic. And so we think the underlying health of the individual makes a big difference.
Just kind of a quick follow-up, and you may have touched on this a little bit. And there's kind of, just out in the world, there's mixed thoughts of people who don't know a lot about what's going on. And we obviously, this is all very new still. Is there any indication on if the antibodies will kind of work the same way as they do with other diseases and viruses? Or, does every virus, do the antibodies work differently? Does that, if you kind of understand what I'm asking, is there a totally new path of antibodies for COVID-19?
That's a good question. The real factual answer is we don't know, entirely, because this is a novel virus insofar as this is a novel coronavirus. There's other coronaviruses, cold viruses are in the same family. And there may even be some protection by what kind of cold viruses you've got in the past, because there's some cross-reactivity. But there's a tremendous amount that we still don't know because of the novelty of this specific coronavirus. Legitimately, our body really has never seen before. But that being said, humans have evolved with viruses for our entire biological history. In fact, a great percentage of our DNA is actually viral DNA, if you want to get completely philosophical. So we've been living with viruses for our entire biological evolution. And so the immune system has been co-adapted to deal with these pathogens. While it's a new virus, it's not like the Andromeda strain, right? That, came from a distant planet, right? This is a family of viruses. Our body has dealt with these kinds of viruses before. And so while novel, it's not unique in that regard. And we have an amazing immune system that is able to, recognize a foreign invader that's truly novel and combat it. And in many ways, most of the symptoms that we have are from our bodies fighting it. It's not really, the virus doesn't have some brain where it has some preconceived desire to kill us or even make us sick. It basically produces symptoms that allow it to spread. And that spreading is what gives it the ability to continue to propagate. So, sneezing, coughing, even the ability for us to, maybe aerosolize as we talk. The virus just is adapted to use those means to spread. And there's even a theory that that perhaps even quarantining can work against some of the strain evolution, that the virus just adapts to whatever is in front of it. So you start to get strain selection that allow it to continue to spread in the face of roadblocks you put in front of it. So the immune system, I think, is well adapted. This is a respiratory virus as far as how it gets into our bodies, although its behavior is very different from other flu virus and cold virus, very, very different the way it attacks the body. So it's very serious in that regard. but it's also a more targeted virus. If you want to think about, it has a tremendously different asymptomatic rate than other viruses like itself. There may be large percentages of the population that have either what's called pauci-symptomatic, which means very little symptoms, that you may not even be aware that you would even suspect you had COVID, or true asymptomatic, where you have like no symptoms at all, or you may be pre-symptomatic, meaning that you have very little symptoms initially and then you get sick later, and that sickness may be mild. And the virus uses this long incubation period to infect and spread around, but our immune system is still combating it all at the same time. And it's probably one of the reasons why, the vast majority of patients have very mild symptoms, and then there's a very targeted, mortality, unfortunately, in patients with these comorbidities, or the elderly. Right now, I think the latest data in Virginia is, if I'm not mistaken, there has been no deaths reported under the age of 25 from coronaviruses reported in the state health department. And of course, I think 80% of the deaths are occurring in people over 70 years of age. So there's this massive dichotomy in who gets it. And I think this is related to, the underpinning health condition of the patient and also their immune system status. So we're starting to get clues about this. We were truly working, from a volleyball term, we were down, 2-0 in a high school game when it began. And I think, we've, I don't know if we've tied it up, but we've won a set. And I think, we're starting to learn every day, every week, every month, we're learning more and more about the virus. And the scientists are learning more, the clinicians are learning more, and we're not completely playing from behind. We're starting to have a toolkit to work from. But the immune system is the body's way to, tie the game up as quickly as possible, because we've evolved this. So, just wrapping up your question, I don't believe that there's going to be some novel, pathogenic mechanisms here. There's always new things. The virus is using known existing receptors that are unique in our body, but we now know what they are, and we're starting to find ways to help prevent that. But there's only so many ways for us to get infected. It's not an infinite number of ways, and we've been seeing lots of viruses in our human evolution. So in some ways, evolutionary, this is just another virus to develop immunity to, either by herd immunity or by vaccination-based immunity. But eventually our bodies will get used to viruses like this, and molecular virologists have said, in years to come, the coronavirus will probably act more like the cold, where it's a nuisance virus. And even with the vaccine, it may just have very mild nuisance-like symptoms, and decades from now or years from now, whenever that happens, no one knows, but that's the usual path. It's not like HIV. You have to understand that it's a good question, because these viruses, like the coronavirus, they don't integrate. They don't integrate into our DNA. You actually have a large population, a vast large number of population of patients recover from infection from this virus. If you think about it conceptually, with a virus like HIV, even though we have good drugs now, once you're infected with HIV, there is no recovery. You live with that virus integrated in your body. It's like chicken pox. We've had chicken pox that people have got infected. It can reemerge later in your life as shingles, for example. So those viruses are very different because they integrate into your DNA. The coronavirus, like other respiratory virus, does not. So when you have this recovery phenotype, that portends good immunity for most all the viruses we've seen like this.
Hey there, thank you so much for watching. If you enjoyed that video, go ahead and give us a like. If you want more content from SciDout every week, go ahead and hit that subscribe button, and we'll see you next time.